Good morning, ladies and gentlemen. Please take your seats and welcome the President of the American Epilepsy Society, Dr. Manisha Patel.
Thank you. I'm delighted to welcome you to the 21st Judith Hoyer Lecture in Epilepsy, Cognitive Comorbidities in Epilepsy, Mechanisms and Potential Therapies. The Judith Hoyer Lecture in Epilepsy combines the discussion of the state of the art in a specific area of epilepsy and the impact it may have on patient care and outcomes. We are proud to partner with the National Institute of Neurological Disease and Stroke in presenting this annual session. We're also pleased to continue to make this lecture accessible to both professional and public audiences. To that end, for the 7th year, we are live streaming this webcast of this public lecture, so many more can listen live and to the archived recording afterwards. I want to acknowledge the support of educational grants from Upshur Smith and the support of the live stream from Jazz Pharmaceuticals. Thank you. And now to a very special award. The AES Extraordinary Contributions Award is designated for those who have made exceptional contributions across multiple fronts, including science, education, leadership, and advocacy. This award is only presented when the Board of Directors determines that there is a candidate whose accomplishments embody the spirit of the recognition. I am honored to present this award on behalf of the AES Board to Mary Ann Meskis. Mary Ann was a founding member of the Dravet Syndrome Foundation in 2009. Prior to accepting the position of executive director in 2012, for more than 25 years, she owned and managed several small businesses in the private sector with an emphasis on project management. This business experience translated her into the role of the executive director. She is a passionate advocate for Dravet syndrome and rare epilepsy communities and has served on various epilepsy working groups and industry advisory panels since the diagnosis of her son with Dravet in 2004. Under her direction, DSF has placed a focus on research as well as patient community engagement and education. As of 2023, DSF has awarded over $7 million in research grant awards, making it the largest non-governmental funder of Dravet syndrome-specific research worldwide. DSF has worked with multiple industry partners on several clinical trials since 2016, including some that are currently underway. These trials have resulted in 3 FDA-approved medications for treatment of Dravet syndrome since 2018. Mary Ann currently resides in Western North Carolina with her husband and the youngest of her 3 children, Elliot, who has Dravet syndrome. Mary Ann, please join me at the podium to receive this well-deserved honor. Congratulations, Mary Ann. The Distinguished Service Award was established in 1993 and recognizes outstanding service by an AES member in the field of epilepsy with a special emphasis on exemplary contributions in service to the American Epilepsy Society, its members, and our patients. I am most pleased to present the 2023 Distinguished Service Award to Dr. Elson So. Dr. So is professor of neurology at the Mayo Clinic Alex School of Medicine. Since joining AES in 1979, Dr. So has served in nearly every area of AES's work, including practice, education, and research committees. Of particular import on behalf of AES, Elson worked with partner volunteer and government agencies to initiate, develop, and advance SUDEP initiatives as a central mission for our professional and patient communities. The SUDEP initiative has continued as a collaborative movement with engagement of more than 30 organizations and has expanded to address the broader issue of increased mortality in persons with epilepsy. An enduring result of this initial effort is the ongoing work of Partners Against Mortality in Epilepsy, PAMI, including this year's record-breaking conference. Elson served as the AES president in 2014, which was a pivotal year for the society. He also served on the Professional Advisory Board of the Epilepsy Foundation and the Council of the American Clinical Neurophysiology Society. He is past chair of the section of Clinical Neurophysiology of the American Academy of Neurology. Elson's research interests are in SUDEP and in the electrochemical aspects of treatments and outcomes of drug-resistant epilepsies. He is co-editor of the book, MRI Negative Epilepsy, Evaluation and Surgical Management. He is named inventor of the Office of Intellectual Property of Mayo Clinic for his role in development of advanced ictal SPECT technologies. He was awarded 2019 and 2021 Teacher of the Year of the Mayo Clinic, Department of Neurology. Dr. So, please join me at the podium to receive this well-deserved honor with our heartfelt thanks for your extraordinary service.
Yeah, just a few minutes.
Of course.
Thank you very much. I'd just like to say two things. First thing, especially to the newer members of the American Epilepsy Society or to those who have not yet joined. We have very dedicated members, leadership and management staff, and AES is a place where we really can advance the science of epileptology and improve the care of the persons with epilepsy. The other thing I would like to mention is that I know many of you have devoted a lot of your time, energy, talents, and resources to the AES. So this award, with which I have the privilege and honor of being associated, is really a recognition and appreciation for all the science that you are doing, for the care that you provide to your patients, and for the service that you give to AES. Thank you very much.
Thank you, Dr. So, and congratulations. Core to AES's mission in the development is the development of the next generation of basic translational and clinical investigators. These young investigators are the future of our community, and they are a vital part of our society. AES encourages young investigators to attend the annual meeting by providing travel and other awards in recognition of the most outstanding abstract submissions. You can see the listing of these early career investigators in the awardee booklet, and I encourage you to visit their posters. In addition, AES supports research through more than $1 million in fellowship and grant awards. An example of the partnership to ensure funding of early career investigators is the AES American Brain Foundation, AAN, Epilepsy Foundation, and the Susan S. Spencer Clinical Research Training Scholarship in Epilepsy. Please join me in recognizing Dr. Dennis Spencer, who will introduce the 2022-2023 awardee.
Thank you. So it's my job to introduce Regan Lemley from the Brigham and Women's Hospital. She is a clinical epilepsy fellow at Brigham's at Harvard, and she studies the role of microbiome in disease-resistant epilepsy. Unfortunately, the planes out of Boston are not very orderly, and Regan is late in getting here. She's not going to join us this afternoon because she's got a delayed flight. And we all know about delayed flights, unfortunately. But let's give her a round of for her this prestigious award. So we know the Susan Spencer Clinical Research Fellowship is a 2-year fellowship, and this is unique because it really provides a course for a young fellow or a young faculty member to be molded for a career, a scholarship career. And it's made possible by the generous gifts of the AES members and others who have made contributions to the endowment that supports this fellowship. It only takes a minute to support the Susan Spencer Fund for clinical research, the Lennox and Lombroso Fund for basic research, or the Sergievsky Fund for epilepsy, health, equity, and diversity. In fact, if you take out your phone right now and you text AES to the number you see on the screen, you're well on your way to supporting any of these positions. Give you a few minutes. Not everybody's as slow as I am with the phone, so. Second, we have a special incentive for donations to the Sergievsky Fund. An anonymous donor has committed to match dollar for dollar each donation to the Sergievsky Fund made between now and June 30th, 2024, up to a total of $300,000. Thank you in advance to all of you who have or will make contributions to help AES support the next generation of epilepsy researchers. And don't forget the seahorse pin at the booths. So, thank you.
Thank you, Dennis, and congratulations to Dr. Lemley on recognition for her research efforts. Before I introduce the chairperson, I want to congratulate an ELC member, Cure Epilepsy, on its 25th anniversary. As one of more than 285 Cure grantees, I have had the pleasure of being nurtured by Cure as a researcher. Cure was established in 1998, and I'm honored to have witnessed firsthand the impact of their grant program makes. The AES is also proud to be the administrative home as well as member of the Epilepsy Leadership Council, a coalition of professional societies, nonprofit patient advocacy organizations, and federal agencies, all working together to expand our impact and improve the lives of individuals living with epilepsy. And now I'd like to invite Gabi Conacher, chair of the ELC Steering Committee, to come to the podium.
Thank you so much, Dr. Patel. It's a pleasure to be here with you all today. I am mom to Elliot Conacher, who is 11 years old and has SCN8A DEE, and I'm very proud to serve in this role as a patient advocate. So thank you so much for the opportunity as ELC chair to share some brief thoughts. As many of you know, the ELC was formed in response to the 2012 IOM report on the epilepsies, which called for an expansion of the Vision 2020 collaborative efforts. So in response to that, a recent community survey that we held, and to facilitate more cohesive epilepsy community by empowering collaboration between our members to identify and fill gaps in epilepsy support, education, clinical care, and research, we are working on some exciting initiatives to facilitate the epilepsy community coming together to partner and we will continue to lift up initiatives throughout the community to advance epilepsy care, treatments, and quality of life, and are working internally to ensure that ELC is an inclusive and representative of all of the epilepsies. The slides you saw when you entered into the hall today, or logged on if you're online with us today on the live cast, showed the work of our ELC member organizations, many of whom are exhibiting in the Epilepsy Resource Center downstairs. I encourage you to visit the booths in the Epilepsy Resource Center and to hear more about the incredible work that all of our members are doing for the epilepsy community. I'd like to thank the American Epilepsy Society for hosting the ELC and providing us with some administrative support to keep our collaborative work progressing. Please stop me in the hallway if you see me, and reach out through AES if you want to be a part of building this collaborative epilepsy community together through the ELC. Thank you.
Thank you, Gabby. And before we move on, I would like to invite Marianne Meskis to give a couple of words right after Gabby Conacher's remarks, if she can come back and, for her honor that she just received. Marianne, are you here? There she is. Please give us a moment. Thank you. This was a concerted effort from our patient advocacy groups.
I thought I got away scot-free. This is not the most comfortable position for me. I don't like to be the one who's in front of everybody, but I just wanted to say a few words. Thank you, Manisha. It's an honor to receive this award. My son Elliot's diagnosis of Dravet syndrome has led me on this unexpected journey. And over the last 20 years, I've been fortunate to see unimaginable achievements unfold and to have the opportunity to play a role in these advancements. Strength comes from unity, and success is not built alone. So I wanted to recognize a few key figures who have brought me to where I am at today. Of course, our Scientific Advisory Board co-chairs, wherever they are in the audience, Dr. Jack Parent and Lori Isom. Not only are they wonderful mentors, but great friends. And Dr. Linda Locks, who diagnosed our son and has supported DSF from the start. Their tireless dedication, along with our amazing staff, board of directors, scientific and medical advisory board members, and all of the other advocacy groups that are here, is just amazing. It really highlights the importance of collaboration and guidance in achieving success. Finally, a special thanks to my husband and my children who've supported this. Thank you.
Thank you, Marianne, and thank you, Gabi, and all of the ELC members and all the advocacy groups for everything that you do. I am now pleased to introduce Dr. Walter Koroshetz, Director of the NINDS. Dr. Koroshetz's presence reflects both the long history and the strong NINDS support for the epilepsy community, as well as his personal commitment to epilepsy research, and we sincerely appreciate his taking time from a very busy schedule to be here with us again this year.
Great. Well, what a pleasure to be here. Schedule's not that busy. This is much more fun than doing emails all day. And it's great to actually see people who are actually doing the work and to talk to families who are affected by epilepsies. As you all probably know, this lecture is named after Judith Hoyer, who was the wife of Congressman Steny Hoyer from Maryland, who we'll hear from at the end of this intro. Judith suffered with epilepsy. She was a strong supporter of epilepsy research and epilepsy care, and also spent her life trying to improve education of children. So a really wonderful person of which this lecture is named. As you know, at NINDS, we are the major funders of epilepsy research. For people with epilepsy, we try to fund investigators that can bring better diagnosis, better treatment, improved quality of life for people living with epilepsy by supporting research across the basic, the translational, and the clinical spheres. We also pay a lot of attention to funding research training and career development programs for the future researchers in epilepsy, and in any instance we have the chance, we promote the timely dissemination of science discoveries and their implications for neurologic health. This is just a graph showing the funding that goes to epilepsy. As I say every year, this is not anywhere near what's needed. That's true about almost all of our diseases. But a lot of the work we do actually cuts across. Some of the basic work is going to be incredibly important to epilepsy. Some of the brain initiative work is very important to epilepsy. The brain initiative is focused on understanding, mapping, monitoring, and modulating circuits, and clearly epilepsy is a clear example of a circuit disorder. So lots of other research would be not included in this, but probably very important. We have been the benefactors of generous funding from the Congress and the taxpayers. Since 2016, we received increases. However, we are been told we don't have a budget now, even though we're spending your money without a budget under what's called the continuing resolution. We're faced with a couple of different scenarios. One, the Senate bill has us with a slight increase of $36 million, which is, well, slight. That means you've been in NIH too long when you call $36 million slight. That's an appreciable increase. But unfortunately, the House bill has us at a $139 million cut, which would be fairly dramatic on the ability to fund new grants. So what we've had to do is to cut our pay line down from 14 to 11 percentile for regular grants. But we're keeping the pay line for early stage investigators at 22%. Now, we're not the only funder, and I want to again congratulate Cure Epilepsy as they celebrate their 25th anniversary. Over these 25 years, they've awarded 285 grants in 18 countries, raised $90 million, funded 290 investigators, 50 early investigators, and hundred thousands of grassroots fundraisers, and created an online community of 100,000 members. And we've at NINDS been really benefited from the relationship we've had with Cure over the years, as well as many of the other epilepsy nonprofits and this society. But in Cure, in 2000, Cure worked with us to establish the first epilepsy benchmarks that is now being carried on by the AES. They became very interested in sudden unexplained death in epilepsy, and there was very, very little research going on in that field in 2008, but we worked with them and hosted the first conference, which led, and eventually, as you see in 2014, to what we call a Center Without Walls project on sudden unexplained death in epilepsy. But the first Center Without Walls was on epilepsy genetics, which had tremendous benefit to the field, which we'll mention in a second. And also, Cure contributed to the creation of the Interagency Collaborative to Advance Research in Epilepsy, where many of the federal agencies get together to share what they're doing and to try to coordinate their activities. So there's been, a really impact of Cure throughout the years. And the Center Without Walls program was so successful that we've, funded one after the other. So the first one, Epi4K, in the genetics, is now morphed into a gigantic international collaborative called Epi25. The SUDEP research has blossomed across the country. We'll talk about some of the products of that initial CWOW that has then spawned on a number of different groups studying SUDEP. And then the epilepsy bioinformatics study looking at the biomarkers that could predict epilepsy after traumatic brain injury and potentially figure out how best to prevent that as one of the modes of actually preventing epilepsy. And then 2 programs which are built to try and understand the causes and the mechanisms of the different gene abnormalities that have been described, either related to channel-related genes or non-channel-related genes. So these are the CWOWs, and I'll say we've been so impressed with them that we now have an equivalent of CWOW for open for any type of research at NIH. It's called an RM1 or combined program, and I'll show you a little bit about that. But just an example of how what we've learned from the CWOWs in epilepsy have affected how we think about doing particularly team science going forward. So the Center for SUDEP Research has had a number of really interesting findings over the years. This one mentioned here from 2021, bringing the idea and some evidence for the idea that there may be a threshold for SUDEP, and it's not just the seizures, but how they occur in approaching this threshold. And then we recently funded a grant to NYU, which is a multi-center grant that will combine and harmonize data from 86 epilepsy centers to study a retrospective cohort who succumbed to SUDEP to see what patterns can be pulled out of that. In the SUDEP field, the group at Iowa published this really interesting report where they recorded from patients who are undergoing epilepsy monitoring, invasive epilepsy monitoring. They found area in the amygdala that when you stimulate it, it would cause apnea, and that this occurs without the person having any sense of air hunger. So their CO2 rises, they should feel short of breath and breathe hard, but they don't. And they also did MRI in this study to find that the amygdala activation led also to activation in the brainstem, the thought being that the amygdala is then driving the brainstem to shut off its sensors and its motor system for breathing. So this certainly brings up potential ways of modulating that connection to prevent SUDEP. There's another example coming out. Actually, comes out of a project at NIH called the Interoceptive Project, trying to understand how we feel from our autonomic nervous system, in this case, breathing. But what they studied was a mutation that causes epilepsy in the mTOR pathway, and in the animals, they were able to show that if you delete this gene in the excitatory neurons, it's sufficient to cause epilepsy and SUDEP in the animals. And in the animals, they have seizures, not all of which are related to cause SUDEP, but the ones that do cause SUDEP unusually have an absence of a theta rhythm that occurs as the seizure is stopping. The non-fatal seizures, you have a theta pattern that occurs as a seizure stops. They don't have apnea, but if you don't see that theta pattern, they do. Again, a clue as to what might be done to recognize these things if this could correlate into the human situation. In terms of the genetic programs, this is kind of the mother of all genetic studies by the Epi25 Collaborative. So they looked at 26,700 people with seizures and 492,000 in controls, and were able to identify 25 genome-wide significant copy number variants. The vast majority of these had not been previously discovered, so leading again to another host of genetic drivers of epilepsy that need to be worked out, and particularly determined to which treatments are best for which types of epilepsy. And there's another example of going down that path, and this is the channelopathy CWOW, and this is looking at a potassium channel, the KCNC2 gene, and they were able to study patients who have this really rare form of epilepsy and implicate the KCN2 gene as the cause. But interestingly, the genetic abnormality causes a whole host of different epilepsy phenotypes and developmental phenotypes not entirely clear how they link up with either the 3D structure of the protein due to the mutations. But again, this is the kind of path we have to keep pushing down to understand these genetic forms and to understand which treatments are going to be best. In this group, valproate, any of those folks who became seizure-free, it was always due to the inclusion of valproate in their regimen, and so clearly some, clinical fallout from these kind of studies. So I can't go through. There's so many things that have been happening in the epilepsy field. I think it's a really robust area of research for NINDS. These are just a couple of the other programs that are either ongoing or just wrapping up that are really important. I put out one, the thalamocortical responsive stimulation for Lennox-Gastaut is actually funded out of the Brain Initiative as an example of a trial to modulate circuits related to epilepsy in humans. And as I've said before, in the Brain Initiative, we've learned a lot about human circuits by studying patients. Not we, but our investigators have learned a lot about studying patients who are undergoing epilepsy monitoring, and then will undergo memory tests or language tests to understand how the brain modulates and functions in those different realms. So the epilepsy patients have been really major contributors to our understanding of human circuits in the Brain Initiative. And just to end up by saying we continue working with the epilepsy screening program, which I think must be around now 55 years. Should check and see when their anniversary is, but it's a program at Utah run by Karen Wilcox, a great program where they have multiple different seizure models. They're very reproducible. They've been doing it for years, and it's open, it's free for people to bring in their assets to test them in epilepsy models. And over the years, it's been very important in developing new anticonvulsants. The push now is on developing drugs that would prevent epilepsy after a seizure in some type of a condition, or to find drugs that work for otherwise drug-resistant epilepsy. So we kind of focused on those two areas going forward. And this is just one recent example about a program that went into the ETSP in 2013 and just recently published positive data from their first-in-human safety biomarker study. So this is probably the latest in those coming out, and hopefully into a phase 2 trial in people pretty soon. So there are a couple of ongoing relevant initiatives. There's a workshop coming up in Rockville where our extramural offices are, June 2021, for genetic treatment strategies for individuals with epilepsy. The epilepsy researchers have been really fantastic in working with us on improving rigor in science, and so there's a prize now. So there's a $50,000 prize for those individuals and teams who can demonstrate that they really did turn things around and improved rigor in their departments or sections. And I just last one is the combined program, and this is a team science program that's open to any type of science at NIH. And as I said, this is really based on the successes of the CWOW that we've seen in epilepsy, and we're trying to spread that out throughout the Institute. We had a recent epilepsy briefing on the Hill, which was really fantastic. Steny Hoyer, who I've seen on the video probably for the last 10 years and never met in person, actually was there, gave a great talk, along with Congressman Greg Murphy and Jim Coster. And it was just a really good event with patient advocates, patients themselves, and investigators. And this is the kind of thing that really makes the Congress folks realize the value of the work that you all are doing. And so I'm pleased here to introduce the Hoyer Lecturer, which the Hoyer Lecturer is Doug Cole, but unfortunately I guess he is not going to be here today, but this is going to be a fantastic lecture about his work. He's a professor of pediatrics and neurology at the University of Pennsylvania, the Children's Hospital of Pennsylvania, and the talk is entitled "The Cognitive Comorbidities in Epilepsy, Mechanisms and Potential Therapies" by Jaideep and Amy. And just to throw out one of the papers that might be relevant is a study that Doug's lab did looking at cognitive abnormalities in animal models with temporal lobe epilepsy, and showed really nicely that if you slow down the hyperactivity in the dentate granule cells, that you can improve the cognition in the animals independent of whether you affect the seizures. So a new idea, I think, that is that in epilepsy there are things related to the seizures and there are abnormalities that are unrelated to seizures, but and we always go targeting the seizures, but we may also have ways, and this is with these chemogenetic tools that came out of the Brain Initiative, to slow down activity, modulate activity in certain areas that could get at some of the comorbidities. So really looking forward to the talk. And now I think we're ready for the video from Congressman Hoyer, represents the 5th District of Maryland for many years. Unfortunately, I think he's talking about retiring. I think that's a bad idea. Retirement is a very bad disease. Thank you.
Hello, I'm Steny Hoyer, Congressman from Maryland's 5th District and the former Majority Leader of the House of Representatives. Welcome to the American Epilepsy Society's 2023 annual meeting. My late wife, Judy, lived with epilepsy, so I personally understand the challenges that can come with a diagnosis. I also saw all that is possible while living with epilepsy. This lecture is a testament to all that Judy was able to accomplish. The AES annual meeting brings together epilepsy professionals in academia, clinical practice, industry, and advocacy from across America and around the world. Crucially, it helps ensure that we keep moving the needle forward to better understand, treat, and even cure the epilepsies. It is inspiring that there are so many patient-centered epilepsy organizations here who collaborate on our shared goals. I'm pleased to see AES's continued commitment to elevating the voices of people with epilepsy and those who care for them within the annual meeting program and in AES broadly. AES remains one of the largest nonprofit funders of epilepsy research, working with its partners to award over $1 million each year to early career and established investigators. I also want to congratulate Cure Epilepsy on its 25th anniversary and to thank its leaders and researchers for all their hard work. Awarding funding to more than 285 cutting-edge projects in 18 countries around the world, your organization is leading the charge to find a cure. Now, before I introduce our renowned keynote speaker for this year's Judith Hoyer Lecture in Epilepsy, I would like to thank Dr. Walter Koroshetz for his leadership at the National Institutes of Neurological Disorders and Stroke. Through his expertise and vision, NINDS is focusing on efforts like the Centers Without Walls, the Brain Initiative, the HEAL Initiative, and the recently announced Community Engaged Health Equity Research in Neuroscience Initiative. I am proud to continue advancing our shared cause here on Capitol Hill. We've made important progress since our last annual meeting. Last Congress, we secured vital funding for epilepsy care and research in the fiscal year 2023 budget. That legislation increased funding for the CDC epilepsy program by $1 million. It nearly doubled from $10 million to $19 million funding for VA epilepsy centers of excellence. It also increased funding for the National Institute of Health by $2.5 billion. Our appropriations for fiscal year 23 included greater funding for a CDC and NIH program that funds the sudden death in young case registry, which can help us get better data and understanding in order to prevent sudden, unexpected death in epilepsy. It's crucial that we protect and increase all of this funding as we work on fiscal year 24 appropriations. In addition, we passed legislation in the 117th Congress to extend telehealth flexibilities so that people with epilepsy can access care in the manner that's best for them. Last year, we also enacted the Inflation Reduction Act, which caps out-of-pocket prescription drug costs for Medicare recipients, more than 1 million of whom have epilepsy. As chair of the Regional Leadership Council, I'm proud to play a leading role in implementing that historic law. This Congress, I was proud to become a founding member of the first ever Congressional Epilepsy Caucus. I thank our co-chairs, Representative Greg Murphy and Representative Jim Costa, for their work organizing the caucus and for their continued leadership to give the epilepsies the long overdue attention in Congress that they so richly deserve. Congratulations to all of you who helped make these changes happen. Sharing your stories and experiences with your congressional office and asking your representatives to join the Epilepsy Caucus are critical to maintaining this momentum. So are leaders like our keynote speaker today, Dr. Douglas Coulter. Dr. Coulter is a professor and neurology researcher at Children's Hospital of Philadelphia and at the Perelman School of Medicine at the University of Pennsylvania. Additionally, he is president emeritus of the American Epilepsy Society. His research focuses primarily on the cellular and molecular mechanisms underlying the development of injury-induced epilepsies, as well as genetically induced epilepsy, autism, and developmental intellectual disabilities. He's been a leader in the field for many years, receiving the AES Basic Science Epilepsy Research Recognition Award, among other accolades. Judy would be so proud to see the progress that he and all of you have made. So let's keep it up. I certainly will here in Congress. I hope you find today's lecture not only insightful, but inspiring. May it help guide your work going forward. Thank you for all that you do.
Thank you, Dr. Koroshetz and Steny Hoyer. Due to unforeseen circumstances, Dr. Coulter was not able to be with us today. Dr. Jaideep Kapoor and Dr. Amy Brooks-Kayal will be presenting the lecture, Cognitive Comorbidities in Epilepsy, Mechanisms and Potential Therapies, on behalf of Dr. Coulter. They have stepped in as former colleagues and researchers in Dr. Coulter's research area and have really done a fantastic job of putting his perspective into this lecture. Dr. Jaideep Kapoor is the Eugene Meyer III Professor of Neuroscience, Professor of Neurology and Director of the UVA Brain Institute at UVA. And Dr. Amy Brooks-Kayal is the Chair of the Department of Neurology and a Professor of Neurology at the University of California, Davis, and both are past presidents. Welcome, Dr. Brooks-Kayal.
There we go. So thank you, Manisha and Dr. Koroshetz, for your lovely review. This, I will say, has been a group effort. One of the things that I love about epilepsy is this amazing epilepsy community, particularly the research community. And when Manisha asked Jaideep and I to do this, we did what we often do, which is we asked for the help of our friends and colleagues in the epilepsy research community. So what you'll see today is some of Doug's research, which we are honored to be able to present to you, but also research from a number of our colleagues who do relevant work in the field. So with that, these are our disclosures. I do a lot of work for NIH, I just want to point that out, Walter, as well as I have a role for ABPN and Annals of Neurology, and Jaideep has also a number of activities that are listed here. Our learning objectives today are basically to better understand the cellular and molecular mechanisms that may underlie cognitive dysfunction in epilepsy, how the hippocampus and cortex work together to store memories, and how epilepsy may disrupt that, as well as understanding the underlying molecular mechanisms that may be the basis for that. So, of course, the most important thing to remember is that epilepsy is more than seizure, something that, fortunately, as a community, we've really come to understand well. People with epilepsy, compared to the general population, have substantially higher rates of cognitive deficits, behavioral and psychiatric disorders, death related to both suicide and sudden death, as well as lower rates of important things like school completion, employment, marriage, and parenthood. And understanding and treating these important components of epilepsy for many people are as important to their quality of life as treating the seizures. And the cognitive issues are not necessarily part and parcel of the seizures. One of the things that we have found is that the cognitive problems actually often predate development of epilepsy. And this is a study from Bruce Herman. Similar studies have been done by other investigators in children and adults that have demonstrated that there are problems with cognition at the time of the initial diagnosis of epilepsy. And you can see here those problems occur in multiple cognitive domains, including memory, but also including language, executive, and psychomotor function. Now, that does not mean that seizures are not in part to blame here, because some of the additional work has demonstrated that over time, children that continue to have seizures and have pharmacoresistant epilepsy have a progressive decline in their development and cognitive functions. So it is really a combination of what leads to the development of the syndrome of epilepsy, as well as the ongoing seizures that seem to contribute to this. And this is work by Ann Berg, and other work has been done in adults that also shows progressive cognitive decline in epilepsy in patients with intractable seizures. So how common is this? Well, hopefully all of us now know this number. 1 in 26 people will be impacted by epilepsy during their lifetime, and that we have approximately 65 million people worldwide who have epilepsy. So those are major numbers. But about a third of those people, or many people believe more, also, in addition to their seizures, have cognitive and or neuropsychiatric comorbidities. So again, we're talking over 20 million people worldwide. We also know that epilepsy has a greater negative impact on health-related quality of life than many other chronic diseases. That anti-seizure medications symptomatically treat seizures, but they do not treat the underlying cause of the epilepsy, and they also do not treat particularly cognitive or neuropsychiatric comorbidities, and can actually worsen them. So how do we go about better treating cognitive comorbidities? Well, to do that, we really have to better understand the cause. And that's where this lecture comes in. So today we're going to talk about the multiple interacting causes, potential causes of cognitive comorbidities as we understand them in preclinical models. So, at the bottom there, you can see a display of multiple cellular signaling pathways, and we're going to talk about the role that changes in cellular signaling pathways have on the development of cognitive comorbidities in epilepsy. Those cellular signaling pathways control gene expression, protein production, and post-translational modifications of protein that result in structural as well as functional changes in neurons, glia, and synaptic function. And those result in alterations in circuit function that probably directly can result in the cognitive comorbidities. Those changes in circuit function and cellular hyperexcitability then feed back and result in additional abnormalities in cell signaling that produce a progressive cycle that can result in worsening cognitive function in patients with epilepsy. So I'm going to start with talking about the molecular and cellular mechanisms, and then Jaideep will pick up and discuss the circuit mechanisms in the second half of the talk.
So when we think about signaling cascades or signaling pathways in the brain, in cells, we think about how critical they are for mediating synaptic plasticity and learning and memory. They are the way that the neurons respond to stimulation, record that, and memorialize it, and result in a physiologic response that changes the way that the next stimuli may be responded to. So this is an example of how learning occurs, for example. Unfortunately, when there's abnormal stimuli, such as a brain injury, seizures, or other forms of abnormal neuronal signaling, those signaling cascades can be excessively activated, and that can result in a pathologic response that can result in network rewiring and aberrant learning and memory. So so as we think about it, the normal balance of activity and signaling cascades produces a balanced physiologic response. Whereas when you have hyperexcitability, such as can occur with epilepsy or after an epileptogenic brain injury, those signaling cascades can be excessively activated, resulting in a pathologic response that has been shown to contribute in preclinical models to both seizures and cognitive dysfunction. And there are multiple, we're talking about multiple pathways. So pathways that are well known, such as the mTOR pathway, and pathways that are somewhat less well known, such as the JAK-STAT pathway, neuronal restrictive silencing factor, and NRF2 pathways, have all been associated with abnormalities leading to abnormalities that lead to cognitive dysfunction. And these are mediated through a variety of molecular remodeling pathways, such as neurotransmitter receptor abnormalities in ion channels, and structural proteins. So just to review a few of these, the one that I know the best because I work on it is the JAK-STAT pathway. And this is a pathway that's the Janus kinase signal transducer and activator of transcription pathway, which is the last time I'll say that because it's long. But what we find is that many brain injuries activate this pathway. And in fact, all epileptogenic injuries in preclinical models have been shown to activate the pathway to some degree. We also know that spontaneous seizures activate this pathway, producing a potential mechanism for the progressive cognitive dysfunction and seizures that can be seen in some acquired epilepsies. And we also know that this pathway is important for a number of things that we think are key to epileptogenesis. And those would include things like neuroinflammation, which is how this pathway was originally described, actually was in the peripheral immune system. But also for things like neuronal differentiation and survival, neuroinflammation, and GABA-A receptor expression, which is how I got interested in this pathway. But it turns out this pathway does a lot more than just regulate GABA-A receptors. It seems to be actually one of the major downstream pathways of brain-derived neuronal neurotrophic factor. And so when you look in neurons and you treat them with BDNF, over 80% of the genes that are regulated by BDNF are regulated through the JAK-STAT pathway. So it's a major regulator of neuronal gene expression. And when you inhibit this at the time of a brain injury, in this particular instance, this is induced status epilepticus resulting in brain injury, you see that there's tremendous activation of the pathway. That's that big, tall bar in the middle. On the left, let's see if I can use, no, yeah, there we go, this one. So you can see there's activation of this pathway, and that you can block that with a small molecule treatment. And when you do that, the animals get many fewer seizures. But importantly, they also have normalization of their memory. So at the bottom is looking at what the animals remember, what arm of a maze they'd been in, something called correct alternation. And what you can see is that normal animals that are not exposed to the brain injury, about 70% of the time remember where to go, whereas once you've had seizures, that drops down, and it can be corrected if you block STAT3 activity at the time of the brain injury. More recently, we've gone on to show that you don't have to do it all over the brain. If you just reduce the STAT3 activity in individual neurons, you get that same sort of decrease that we saw in seizure activity. with the red bar being the number of seizures in the animals that had the reduction in STAT3 just in their neurons. And you also see that they don't develop as many seizures. But most importantly, what they also don't develop is epilepsy-associated memory deficits. So when we looked both at motor learning with a rotor rod and fear conditioning, which is a hippocampal memory function, contextual fear conditioning, we saw that unlike wild-type animals that had developed epilepsy who could not remember when they had been in a context that was dangerous, we found that animals that had a knockout of STAT3 just in their neurons had normal levels of memory. So suggesting that this may be an important potential pathway to regulate, to improve cognitive function, even after the onset of epilepsy, because again, these animals had, we were blocking the STAT3 well after they had developed epilepsy. We also found that this seemed to be related to alteration in gene expression. About 3,000 genes are altered in the hippocampus after a brain injury like interhippocampal canic acid. And STAT3 regulates over half of those genes. So when we knock out STAT3 just in neurons, we can correct the abnormal gene expression at about half of the genes that were abnormally regulated. And importantly, many of these pathways are related to learning and memory. So again, key regulators of cognitive outcomes once people develop epilepsy. Now, the good news is that the STAT3 pathway has a small molecule or drugs that are currently available that regulate it. None of them yet are really designed to be used in the central nervous system, but they exist and we know they work. So again, a potential small molecule that could be developed that would potentially improve cognition. Another pathway where there are therapeutics that are currently in existence is the mTOR signaling pathway, which I'm sure most of you have heard about. That pathway is critical for cell growth and proliferation, for dendritic growth and protein synthesis, regulates neurotransmitter expression, and all of those, that translates to an effect on learning and memory and social behavior. This is a pathway that is abnormal in a number of epilepsies, including a number of genetic epilepsies, such as tuberous sclerosis complex and DEP-DEC5. It's also altered in acquired epilepsies like hypoxic-ischemic encephalopathy leading to epilepsy, infections, and a number of preclinical acquired epilepsies that both post-traumatic and the chemoconvulsant epilepsies like pilocarpine and canic acid. So we know we have a current medication, rapamycin, everolimus, is used in patients that we know blocks activity in this pathway. And a number of labs have shown that if you block this pathway, you can impact seizures in preclinical models. So this is work by Mike Wong's group that showed a number of years ago that when animals are exposed to canic acid-induced status epilepticus and develop spontaneous seizures, that they have much higher levels of phosphorylated S6, which is a marker of activity of this pathway. And that if you block that with rapamycin, that you can substantially reduce the number of seizures that these animals have. And that's the animals that have epilepsy without the rapamycin treatment are in the black bars, and the smaller gray bars are the animals that were treated with rapamycin. But importantly, and more relevant to this talk, it also has a very positive effect on cognition. So this is work that Amy Brewster did when she was in Ann Anderson's lab. And what this shows is that animals that receive rapamycin following their status epilepticus have improved cognition and memory. This in particular, on the left, you can see that the animals with epilepsy alone have spend less time in the correct part of the maze, in this case the Morris Water maze, compared to animals that are treated with rapamycin following status epilepticus, who look basically the same as the control animals. So Amy went on to look and show that this seemed to be related to changes in dendritic structure. So the two on the left are the control animals, where you can see the normal dendritic structure. Animals that have been exposed to status epilepticus and developed epilepsy, on the third panel there, have a substantially disrupted dendritic structure, and that's pretty much normalized in the animals that receive rapamycin. Mike Wong's group went on to show that the change in the dendritic structure was associated with loss of dendritic bulbs. So as you can see there, there's presynaptic spines on the top. We're going to try this again to see if I can do it without advancing the slide. There we go. These are the control animals, either just got vehicle or got rapamycin. You can see when animals receive canic acid status epilepticus, there's a substantial reduction in the number of normal dendritic spines, and that when you give rapamycin, that is substantially improved and brings the animals with epilepsy up much closer to the control animals. So again, the pathway causes changes in proteins that result in change in dendritic structure. And then Tali Barham's lab has really led the way on looking at the neuronal restrictive silencing factor pathway, also called the rest signaling pathway. She's been particularly interested in the febrile status epilepticus model in rodents that results in both epilepsy and cognitive dysfunction. NRSF is involved in controlling gene signaling through the HDACs, and her lab demonstrated that there was a broad repression of gene expression following febrile status epilepticus that could be normalized so this is the broad repression and this is the normalization when she blocked NRSF signaling with antisense oligonucleotides. She also went on to show that blocking NRSF both reduces the number of seizures the animal gets and also substantially improves cognition after febrile status epilepticus. And then went on to demonstrate that was being regulated actually through metabolic demand in SIRT1, which resulted in a change in miR-124 and increased NRSF signaling. And through that work, they have now developed a small molecule that they're using in this model to see if they can effectively reduce NRSF signaling and improve memory deficits and potentially reduce seizures after febrile status epilepticus. And then this important area, metabolic signaling in epilepsy, has been led by my colleague to my right, Dr. Manisha Patel, our president, who's shown in a series of studies that epileptogenic brain injury results in increased mitochondrial reactive oxygen species that leads to both bioenergetic dysfunction and post-translational modifications. And that mitochondrial dysfunction seems to be involved both in the development of seizures, neuronal injury, and cognitive dysfunction. She, with the work of members of her lab, demonstrated that if you use a small molecule metalloporphyrin, that you can if you reduce the reactive oxygen species there in the bar to the yellow. So this is following the treatment with the small molecule that reduces reactive oxygen species. You can improve mitochondrial respiration, reduce neuronal loss, and improve cognitive function. So again, suggesting that there may be multiple ways that we can improve cognitive function following brain injury and epilepsy. Most recently, Tim Simeone has done work also looking at mitochondrial function to improve cognitive function, which apparently is what nootropic means. Tim sent this slide to me, and the first thing I had to do was Google nootropic. So if you don't know what that means improves cognitive function. So he's used a KCNA1 genetic model of temporal lobe epilepsy in mice to show that in these mice with epilepsy, they have mitochondrial dysfunction, decreased mitochondrial respiration, reduced ATP production, and excessive generation of mitochondrial reactive species. And they have impaired cognition and long-term potentiation and abnormal hippocampal network oscillations. What he also has found, most importantly, is that by using the ketogenic diet or even ketone bodies themselves, he can restore memory in this model, restore LTP, and normalize hippocampal oscillations. And he can do that, in addition to doing that with dietary therapy, he's also shown that he can do that with a mitochondrial targeted small molecule antioxidant, again, suggesting that there may be many ways to potentially modify these pathways. So as far as this brings us to the end of the signaling, cellular signaling part, but just as an overview, luckily it seems like there may be many ways that we can potentially reduce abnormal signaling following epileptogenic brain injuries and the abnormal signaling that is maybe incited by spontaneous seizures themselves. And that if we can normalize that either with small molecules that target any of the several pathways that we just discussed, or through dietary or small molecule treatment that improves metabolic function, we may be able to reduce that pathological response and decrease cognitive dysfunction to improve the lives of people with epilepsy. And for those of you who have an interest specifically in cognitive comorbidities and also in the importance of metabolic signaling in that, I would recommend that you attend the Dialogues in Epilepsy session on Monday, which the two topics are going to be mechanisms of comorbidities and also and metabolic function. So with that, Dr. Kapoor, I think you are up.
So that's a tough act to follow. So I'm going to talk about circuits and how circuits modulate memory. The founding paper on memory, and I'm going to focus on just one cognitive comorbidity, that is memory, and specifically memory of what, when, and where. And that is coded in the hippocampus, and that was seminal contribution of epilepsy research to memory research. This is HM, or Henry Molaison, now that he's passed away, who underwent bilateral temporal lobectomy by Scoville, a neurosurgeon in Connecticut. And after that, he never remembered anything new. And Brenda Milner, shown here, studied him over his lifetime. And no new memories were formed by HM after that. And we know that that was the first clue that medial temporal lobe, or the hippocampus, is deeply involved in memory. I'm going to be talking both about mouse and rat hippocampus, which is shown here, and the human hippocampus, and try to show you that advances that we are making in understanding hippocampal memories also apply to human hippocampus. And one of the fun ways of showing this is, I love this paper. There are hundreds of papers like this. This is a cab driver in London who's put in a fMRI machine and asked to imagine his route from Elephant and Castle to Grosvenor Square. Now, he's just imagining the route, and what happens is that his hippocampus lights up. There are many ways. You can do it with EEG. Many of our patients with intracranial EEGs undergo similar kind of testing, and you can show with memory, but it's a fun way of seeing that the hippocampus lights up when you have to recall spatial information. So what is the hippocampal circuit? Since I have to talk about circuits here, hippocampus receives most of its inputs from internal cortex. Internal cortex is probably the best connected cortex in the whole brain of all the cortices. It seems to receive most of the information both internally and from external senses, and that's funneling the information to the hippocampus. And as I'm talking to you, this information is being sent to your hippocampus, and our goal is for you to remember some of what you hear today. And this feeds into the next structure, dentate gyrus, and I'll be speaking a lot about that, which then feeds into CA3 and CA1 hippocampus, and I'll talk a little bit about the CA1 hippocampus. There is a feedback into neuronal cortex from the hippocampus, and some of it is reentrant circuit, which can also helps with memory, but also creates challenges. So one of the first discoveries, and this won a Nobel Prize in 2014, was made by O'Keefe in London. And what he noticed was that in CA1 pyramidal cells, if you put microelectrodes in, and a rat or a mouse moving through a maze, cells would fire when they were in that maze. And I can say personally that my first experiment that worked as a graduate student way back was to record place cells, and it's a religious experience to see that day after day, week after week, and we were doing a paper on stability of place cells, the cell would fire at the same spot on an 8-arm maze. But then Dr. O'Keefe made another discovery, which is seminal to memory literature, and this is called phase precision. This is a bit complex, but I'll try to explain to you. So if you record local field potentials, or EEG, from the hippocampus, what you get is a 4 to 7 frequency oscillation called the theta oscillation, shown here. And as any oscillation, we divide it into 0 to 360 degrees. If you look at the place cell firing shown here, you can see that the place cell firing is occurring at a specific phase. And as the mouse proceeds and more theta waves go through, the next time the firing occurs somewhat earlier than when it occurred. So here it is near the peak, second time it's below the peak, third time it goes further away from the peak of theta oscillation, then it is still further away from theta oscillation, and as much as 350-degree shift can occur in firing. And this is sequentially reproduced in place cells that are covering a space. So if you have a linear maze and you have a mouse or a rat run on a linear maze, you'll have that sequential theta phase precision. So why is that important? Because you not only want to know where you are, but where you're going and what's going to be the next thing. If you couldn't predict where you are moving to, you would get lost, like I did this morning coming to this hall, because I had never been here, right? But what happens in epilepsy, and this is a paper from Greg Holmes' lab, Langsantini and Holmes in 2008, made very important observation in epileptic rats. They again studied phase precision. Again, this is the normal phase precision, as I showed you in previous slide. And many of the cells still had normal phase precision. But when many other cells showed no phase precision at all. So, and they quantified the number of cells that were not processing spatial information, and that was 35%. So this is important information that spatial memory encoding itself and is disturbed in epileptic rats or mice as well. Another important discovery made in 2004, replicated many times since then, made by where the internal cortex cells form grids, and these grids literally make a grid of the space. For example, if you want to have a GPS, you want to have a grid in which your place cells are firing. And Mosier made this discovery in 2004, and these have been confirmed many times. And not only do they form a grid, but there is phase precision in theta phase precision even in these cells. So these cells are feeding information that is then being interpreted into place cells in CA1. But there is an important structure that lies in between them. That's the dentate gyrus and dentate granule cells. The dentate gyrus attracts a lot of attention in epilepsy research, and I'll show you why that is. First, to understand that dentates function, there are many theories of how dentate gyrus is working. Classical theory is that it is a sparse coding structure. That is, a lot of entorhinal cortex cells may be firing in different patterns. But what happens is there are far more granule cells than there are entorhinal cortex cells. So a single pattern of firing shown in input 1 or input 2 would lead to one set of granule cells to fire, a very few of them of the total, while second set of inputs will cause a different set of granule cells to fire. There is consensus that this is what happens, or what we call sparse coding. But in the epilepsy field, the theory that really takes a lot of interest is dentate gate hypothesis, where the idea is that dentate serves as an inhibitory gate, preventing seizures from entering CA1 and moving on. And this dentate gate, a lot of work done by many, many investigators in epilepsy field have shown that this dentate gate is due to strong inhibition, low excitability of granule cells, and other mossy cell circuits. So why are we all so interested in dentate gyrus? You'll see this from this types of temporal lobe epilepsy. As many of you know, temporal lobe epilepsy is the most common form, or one of the most common forms of epilepsy, especially medically refractory epilepsy. And if you look at human temporal lobe, this is a normal temporal lobe. You can see dentate gyrus, CA3, CA1 neurons, subiculum. But if you take patients with temporal lobe epilepsy and who underwent temporal lobectomy, you'll see severe loss of cells in CA1. That's one pattern. In some patients, there is a sclerotic hippocampus where all the CA fields are gone. And in these patients, it's very easy to understand that they have memory problems because they've lost cell layers. But then there are patients, many patients, who have minimal cell loss, and that's mainly in the dentate hilus, or the cells that interneurons that lie between two blades of the dentate gyrus. Yet they have refractory epilepsy and memory problems. And question is why? If this is the minimal lesion that causes temporal lobe epilepsy, how does it do it? And one of the important findings in this field was made by Amy right here with Doug Coulter, which is probably why we're here, they showed that the GABAergic inhibition of dentate granule cells is altered in a specific fashion, which has been reproduced by, I think, 50 papers since their original 1998 paper, showing altered GABAergic inhibition of dentate gyrus. The second set of observations that again has been repeated by many, many labs, starting with Bob Slovater, is that interneurons in the hippocampus are dead. I just noticed that the slide is flipped. And these neurons, the interneurons, inhibitory interneurons die, and this reduces inhibition. And this led Dr. Coulter to propose that the dentate gate, as I just talked about, with reduced inhibition, would be altered. So here, this is a slide, and Walter also showed this slide just before me. This uses TRAP2 animals. TRAP animals express a red protein called TD tomato when neurons are active. I won't go through the whole genetics of this, but I'll tell you that this is one of the great achievements of the Brain Initiative, developing these novel mice, that we can see every cell that's active. So everything that's red is a neuron or a glial cell that's active. And in control, as Doug's lab shows, that dentate is not very active, as I showed you earlier. It's a sparse firing structure. In an epileptic animal, a lot more dentate granule cells are active. Right at even though there's no seizure at that moment, even without a seizure, the dentate's filtering function is disturbed. Of course, after a seizure, you see bright granule cells light up, but the important point is in between seizures, this function is altered. What dentate appears to do is to help us discriminate between objects in locations. So Doug's lab designed this very simple paradigm for testing spatial memory, where you put 3 objects and ask, and after training, you remove the mice or rat, come back after a period of time, and only one object is moved from original location. A normal mouse will explore this novel location because it says, oh, this was not in this place before, so the normal mouse will explore this. But an epileptic mouse appears to ignore this, and this is the spatial deficit that his lab set out to fix. Now, another one, and Walter mentioned it in his talk, is how to do it with chemogenetic methods. So one of the chemogenetic methods is called DREADS, designer receptors exclusively activated by designer drugs. And what you can do is put these designer drugs into granule cells, which is what they did. They added a color marker so they could see where their viral delivery of that drug was, of these receptors was, and then activate or inhibit using these drugs. And in the next slide, they show the effect. And what Doug's lab showed was that if he inhibited some of the neurons, he could improve the performance, and the mice started behaving and discriminating the location of the novel object shown here just as well as controls. But if he did it too much, right? So if he inhibited most of the cells, what happened was the opposite. The mouse became stupid again and did not do this discrimination task. There's some technical concerns with this drug, CNO, so he had done it with cord, but this is repeated with 2 different chemogenetic agents. Again, very rigorously replicated experiment that by fixing the filtering function of the hippocampus using chemogenetics, he could improve memory. Another work, and I'll highlight some of it, is work by Tristan Schumann, and where they were looking again at the timing of interneurons in dentate granule cells, dentate gyrus. I talked about these neurons being affected in epilepsy within the dentate gyrus, so they put these very sophisticated microelectrodes with multiple contacts, and then looked at epileptic and control neurons. And again, these interneurons also fire in a phase lock fashion as it relates to the theta rhythm. So theta phase locking in a normal interneuron, and then shown here in control, you can see very specific phase lock firing of interneurons in memory. But when you go to epileptic neurons, this is all over the place. So it's the phase locking itself has been altered. So what Tristan's been doing, and this is unpublished data that he shared with us, is to see if, this is again in CA1, I'll just go to the fixing part because I've shown you the C1 data, is what using optogenetics. Now these are again tools developed with the Brain Initiative. Here, instead of using chemogenetics, these are molecules that react to light, and they open in response to light. They are opsins. But because this can be done very precisely in terms of time, you can deliver your stimulus or optogenetic stimulus at a specific phase of theta rhythm. And this requires a bit of computer programming and good amount of coding so that you get it right. But if you do it, then you can, so the programming is to make sure that you're getting the theta rhythm accurately and then firing. And then the idea is we can fix the interneuron firing and get it timed right and then improve behavior. So all this theta rhythm work I've talked about is in the mouse hippocampus or the rat hippocampus, but does it really happen in humans? And there's plenty of data, and this is just, I'm showing you one slide which is a review from Kahana, Mike Kahana at Penn, showing that both spatial navigation coding and list learning in humans, you can talk to people and you can make them remember. And those, he's shown that those are coded along the theta rhythm. And again, one of the most important things, as you read the literature on human memory, many of these papers get done on our epilepsy patients because they are the ones with hippocampal electrodes, and they are the ones where they stay with the hippocampal electrodes in our epilepsy monitoring units for days when this kind of testing can be done. One last thing that just to remind me and everyone that hippocampus is not the beginning, maybe the beginning, but not the end of the memory. Memory consolidation occurs because these hippocampal rhythms coordinate with cortical rhythms, and there is what we call a process of memory consolidation. That is, the memory moves from the hippocampus to structures like retrosplenial cortex, mediodorsal thalamus, and then on to medial prefrontal cortex. There's a whole complex process of memory consolidation. And the coupling between the hippocampus and the cortex is disrupted by interictal epileptiform discharges. These are the epileptiform discharges that are occurring between seizures, and as Amy pointed out to you, the cognitive deficits can occur between seizures as well. Many of them we now believe are related to epileptiform discharges. And this paper by Jennifer Galinas, who's talking at one of the SIGs, I think, during this meeting, shows that disruption. It's a much bigger story, but I just wanted to mention it at the end. So let me end by showing you this slide that Amy showed earlier. You can understand cognitive comorbidities at multiple levels, as she pointed out. You can look at cell pathways, molecular pathways, genomic expression. You can look at it at synaptic transmission or cellular excitability level, and you can look at it at circuit level as shown. And there is hope, and there's important progress being made at each level, which we can say would make a difference to our patients' lives eventually. Some of these things like ketogenic diet and others may be available very soon. Things like precise optogenetic timing of deep brain stimulation may take time, but I'll remind you that we use deep brain stimulation for treating epilepsy already. We have about 16 years of experience. So I'll stop there and thank you.
I want to thank Drs. Brooks-Kyle and Dr. Kapoor for doing an outstanding job of summarizing Doug Coulter's body of work in his research and an area that's so dear to Doug, the cellular mechanisms of the comorbidities of epilepsy, and just leading to the potential therapeutic implications that this work has. I want you all to have a wonderful conference, and thank you so much for your attendance. And this ends our Hoyer lecture. Thank you.